Gastrulation, formation of the primary germ layers. Gastrulation occurs in an embryo during the third week of development, during which the bilaminar embryonic disc is converted into 3 primary germ layers, the sources of all tissues in the embryo. This part of the implanted blastocyst shows the embryonic disc sandwiched between the amniotic cavity and umbilical vesicle. Note the epiblast, the hypoblast, and the prechordal plate. Let's look at the surface of the epiblast, which shows the location of the oropharyngeal membrane and the cloacal membrane. By the end of the second week, the primitive streak, a thickened band of epiblast cells, appears in the caudal portion of the embryonic disc. The next few transverse sections of the embryonic disc show how the primitive streak is formed. Cells from the deep surface of the primitive streak are transformed into mesenchyme. The intraembryonic mesoderm is derived from some of this undifferentiated mesenchyme or mesoblast. Cells from the epiblast and primitive streak migrate into the median plane of the embryonic disc to displace the hypoblast. These cells become the embryonic endoderm, which forms the roof of the umbilical vesicle. Cells remaining in the epiblast become the embryonic ectoderm. As the primitive streak elongates by producing cells at its caudal end, the cranial end proliferates to form the primitive node. A depression or primitive pit appears in the primitive node, which attaches to a groove that develops in the primitive streak. The notochordal process extends from the primitive node to the oropharyngeal membrane. It forms as a result of cranial migration of cells from the primitive node. Mesenchymal cells from the primitive streak migrate laterally and cranially, forming the mesoderm between ectoderm and endoderm, except at the oropharyngeal and cloacal membranes. Mesenchymal cells reach the margins of the embryonic disc and are connected to the extraembryonic mesoderm covering the amnion and umbilical vesicle. The primitive streak regresses toward the sacrococcygeal region, degenerates, and usually disappears by the end of the 4th week. Observe how the notochordal process is transformed into a solid notochord. The floor of the notochordal process fuses with the embryonic endoderm, which eventually breaks down and disappears. The remnants of the notochordal process intercalated to the endoderm form the flattened notochordal plate. Cellular proliferation and infolding of the plate result in the formation of a solid notochord, which separates from the endoderm. The notochord triggers the overlying ectoderm to thicken and form the neural plate. The neural plate invaginates in the midline to form a longitudinal neural groove with neural folds on the sides. The neural folds merge and form a neural tube, the primordium of the brain and spinal cord. The notochord defines the longitudinal axis of the trilaminar embryonic disc. It is an important signaling center in the primordial embryo and plays an essential role in the formation of the central nervous system and musculoskeletal system. The notochord then regresses, but remnants, referred to as the nucleus pulposus, contribute to the formation of the intervertebral disc. The allantois is a small diverticulum extending from the caudal part of the umbilical vesicle into the body stalk. In the embryo, the proximal part of the allantoic diverticulum is known as the urachus, which extends from the urinary bladder to the umbilicus. It becomes the median umbilical ligament in the adult.